The Cancer Genome Atlas, as you can see, the letters come just from that, the Cancer Genome Atlas, TCGA. It was launched in 2006 as a pilot project and was so phenomenally successful that it was expanded in 2009, and it just ended with 33 cancers overall undergoing what I'm going to describe to you as a comprehensive, and I want you to keep these words in mind, I'm going to keep coming back to them, comprehensive, integrated analysis of key genomic and molecular features in, as I said, many cancers, 33 total. And now there's a lot of integration going on even between cancers now that we've gone deep in single cancers. This was an NIH-funded project, and the goal of each of the individual projects was to publish, as Sarah mentioned, a marker paper. And the marker paper is the fundamental discoveries from the project. But importantly, in addition to that paper, which is available to everybody free, that the data from the project would be made publicly available to researchers throughout the world, so that this was not a closed project at all. This was meant to be extremely open for any researcher to look at this data. And it really serves as a model of the power of teamwork in science. You've heard about teamwork a lot, and certainly everything that's going on that you've heard is a testament to this. But in this case, it was really a worldwide team effort. We had folks from the UK, France, the Netherlands, Canada, and a whole bunch of groups in the United States. And this was because we had to get the best samples that we could have the most quality control on to make sure that, as you've heard the saying, garbage in, garbage out. We didn't want to come out of this not really learning anything because we started with insufficient materials, et cetera. And you can imagine the coordination that's involved. And as I mentioned, uveal melanoma was one of the rare cancers chosen, and this really is a testament to the advocacy of MRF and CureOM, because at the time they said, look, we have some extra funding, we're interested in doing rare cancers, and because of what they were hearing from CureOM, they decided to take us on. This is a lot of negotiation that went on, and I think on the website you document that with Kenneth Shaw from the NIH and how that process occurred. And so it's a point that has to be made, is that all of these individual efforts that you talked about earlier really bubble up into this multi-million-dollar effort ultimately to examine uveal melanoma. And so getting back to these words, comprehensive, integrated analysis of features. So you're all probably familiar with this cartoon where you have each of these investigators. They have, they all can't see because they have a cloth around their eyes, and one is grabbing the tail and one is grabbing the trunk, and they all are thinking that they're finding, the key feature in what they're looking for. But of course, they only have parts. And so by comprehensive, really meaning all the features, and integrated, meaning how do they go together? The tail doesn't connect to the nose. We have to better understand how each individual feature, and I'm going to describe what I mean by feature along the way, how each of these individual features, if we take all of them that we can get our hands on, how they interconnect and work together to drive this cancer so that we can identify key Achilles heels to target by different mechanisms. And it was said earlier by Kim Roy, knowledge is power, absolutely the case. And I'm giving myself this quote. I hope I'm not quoting somebody else, but there's not a principal investigator. There's not one senior researcher of the Mars rover mission, right? When you think about it, there's the team that did the fuel, and the team that did the communications, and the team that set up the computers, and that's really ultimately where we're going in biology, I think. To do major projects of this type, we're going to need people with skill sets that just cut across the board, and no one person is going to be necessarily the Einstein. We're going to have a whole bunch of, hopefully, Einsteins on board. So, okay, now let's put our biology hats on. Okay, I'm going to do a little bit of primer here on biology in 60 seconds. So, DNA, as you know, the double helix, is made up of these chemicals, cytosine, guanine, adenine, and thymine. That's all it is. And we we list them as letters, C, G, A, T. And so it's just a series of C, G, A, T's in different order, T, 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 A, T, C, G, T, T, C, D, T, T, right? All along, right? And the way I like to think about it maybe a little bit kind of is like the old telephone book, right? You have the old telephone book with just numbers. Numbers. In this case, there's only 4, and they're in different orders. And then some set of them is actually a phone number that links, that does something. The rest of them do other things we're figuring out, but there's some set of phone numbers that actually make the call, okay? And so we call those genes among them. And we have about 20,000, maybe 22,000 genes in our whole genome. So a gene is a specific set of those A, C, T's, and G's, a certain code, and the genome is all 3,200,000,000 of those A, C, T's, and G's, okay? And so the genes only make up 1% of the whole genome. Only 1% of the phone numbers in that stack of phone books makes the signal, all right? We call those genes. And so when you wrap up all of these C, G, A, T's, the double helix, when you wrap them up, you get a chromosome. And that's how DNA is stored. It's stored as a chromosome. And you have 23 pairs. So you have 23 from mom, 23 from dad, get together, 46. And they're pairs. So you have 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s, right on down to an XX or an XY, depending on what happened, right? So, and then that is in your nucleus. So your nucleus has those 23 pairs of chromosomes, and when you unfurl them, you have about 20,000 genes, okay? And genes can be regulated by multiple things, but one of the things that genes can be regulated by is what's called methylation. And that's simply just think of it as an on-off switch. And most of the time, there's always exceptions in biology, when you're methylated, it means that the gene is not going to be expressed. That phone number is not going to be called. Versus when you're unmethylated, it allows for the gene to be expressed. And so it's not this simple, but just for our purposes, methylation off, non-methylated on, and this will come back. This is a feature. This is one of the parts of the elephant that we did a deep, deep investigation on, and I'll talk more about that. What are some other features? Well, I mentioned the DNA, the genes, and like I said, the ones that can actually make the phone call get transcribed into what's called messenger RNA, the message, because it's a code, and then that gets translated into a protein. And we're familiar with tons of different proteins, insulin, collagen, many different proteins that came from the information in DNA via the transcription and then translation of mRNA. So this is another feature, genes, mRNA, another feature. So we could look at all the transcripts, all the mRNA in the cells of the tumor. We could look at every gene in the tumor. Comprehensive. That's what I mean by comprehensive. We're going to look at every gene. We're going to look at every mRNA, and some of the RNA isn't translated into protein. They're called microRNAs or link RNAs, and they have different functions that I won't get into, but the point is, again, comprehensively, we're going to look at all the miRNAs. We're going to look at all the link RNAs. Comprehensive look at all these different features in the tumor through sequencing. So another feature that we could look at is what we call chromosome number, or it's also called copy number. And actually, uveal melanoma, if I was teaching a class of medical students who were going to go on to do all kinds of different things in their lives, I would use uveal melanoma as a classic example of copy number change in cancer. And you are all familiar with these words, and I'm going to try to drill down and show you what they mean. So disomy 3, monosomy 3, BAP1. These things are related. In what way are they related? Well, I told you, you each have 23 pairs of chromosomes, so 46 total. You have 2 chromosome 3s in your normal cell, 2 chromosome 3s. And chromosomes have a small part called the P, you can remember that by petite, and a big part, the Q. So every chromosome has a P and a Q, and you have 2 of them, one from mom, one from dad. And the normal situation is disomy 3, 2 chromosome 3s with 2 Ps and 2 Qs. So that's the normal state. In uveal melanoma, within the cancer cell, you weren't born with this unless there was a germline issue, which we won't talk about at the moment. You weren't born with it. It happened in the cancer cell. You have a loss of one of those chromosomes 3. Thus, we call it monosomy 3. Which one did you lose? Did you lose moms? Did you lose dads? Actually, I don't think many people have studied that. You could. But at the end of the day, you have one chromosome 3 in the melanoma tumor cell, okay, versus the normal. And so how does that relate to BAP1? You heard about BAP1 mutations. Well, guess what? BAP1, the gene, is one of the genes located on chromosome 3 on the P. So in the normal situation, you have 2 BAP1 genes located on 3p21 to be exact. And in the case of uveal melanoma, for about half of patients who are monosomy 3, you have a loss of chromosome 3, thus a loss of that BAP1 gene because it was located there. And on the remaining chromosome 3, you have a mutation in the BAP1. So therefore, you've lost both BAP1 genes. One because you lost the whole chromosome, and one because you had a mutation in the remaining chromosome gene, BAP1, okay? And that's what it means. So when I'm going to use the fancy word somatic copy number alteration, somatic meaning you weren't born with it, and then copy number meaning how many of chromosome 3s you have, alteration. So this would be a loss of chromosome 3, monosomy 3, with a coterminous BAP1 mutation. Okay, and this was identified by Bill Harbour in 2010. All right, so that gives you some definition on some features, genes, chromosomes, RNA, and let's go over them. So we talked about genes. Genes can be mutated. I gave you the example of BAP1. Genes can be methylated. That can change their expression. Chromosomes, you can lose parts of chromosomes. You can gain parts of chromosomes. I talked about a loss, as in monosomy 3. RNA comes in different varieties, microRNA, lincRNA, and then the messenger RNA, which goes on to make protein. So these were the key molecular features that we were going to look at every single one of these that we could identify of these features within uveal melanoma. And couple that with a comprehensive analysis because we also had, I'll go to the next slide, we also had the hard work that had been done by multiple groups to one, bank the tumors in the right way, have them be frozen, have clinical annotation of them, know features about the pathology, how big it was, the diameter, whether it was epithelioid, et cetera, et cetera, et cetera. And also clinical data, how well patients did, if they went on to get therapy. In some cases, we had that kind of data, whether they had recurrence locally. And so all of that happened over about a 2-year period of just figuring out who has the right kind of tumors that is not going to be garbage in, that has all of this wonderful clinical data, pathological data, that we could test all of these molecular features comprehensively on, and then in the end, integrate. Integrate by saying, well, if you have this profile, does it correlate with anything clinically? If you have this copy number alteration, does it connect at all? And that's what I'm going to show you today is some of the connections, the integration we did by comprehensively looking at all of these molecular features along with clinical and pathological features. And you can see these things, this was run incredibly well by the NIH, I have to say. And they had deadlines and they had a huge team of people that once our pathologists gave QC, they QC'd on their side. They had to QC to make sure the DNA wasn't degraded, the RNA wasn't there, it was all in one great shape. They did all the sequencing at multiple sites, which is no small task. They gave themselves deadlines to get these things done. And then we got it, and we were tasked with make sense of it. And we had a huge computational team, clinical and molecular group of folks. And they gave us 2 years, get that done. And if anybody knows how, unfortunately, this business works, it can take a year just to get something published once you get it done, especially in a very good journal where it's going to get recognized. And so it took us actually 3 years total, but we got it done. And like I say, this was all because of a lot of bubbling up of the kind of work that is done right here in this room to push for it so that we could get NIH to take us on. And subsequently, I'll jump to the chase. Because of that, there's actually a new effort that's going on to look at further features in metastases, and we've put in our bid for uveal melanoma. So that's coming up. So, like I mentioned, these are the features. We're going to look at copy number. We're going to look at every single one of those 3 billion nucleotides. We're also going to look at every one of the genes even more deeply. we're going to look at mRNA, microRNA, link RNA, DNA methylation, protein, and then we're going to integrate it based on clinical data and pathway analysis. So this is the first, this is what we actually look at. So the data comes through, we have a computer say, okay, there's 80 samples. We had 80 samples from France, England, United States, and a few other places. And then these were the chromosomes I mentioned, 1 through 22, plus the sex chromosomes XY. And then they were either P or Q, PQ, PQ, PQ. So as you look, it's 1 PQ, 2 PQ, 3 PQ, 80 samples. And blue means that you have a loss from the normal 2. Red means you have a gain from the normal 2. White means you have the normal 2. You can tell right now what the computer did. The computer just says what sample is more like every other sample. These samples look a lot more alike than these samples. These samples look like each other versus these samples. So in an unsupervised way, the computer does this, and then we step back from it and say, does it make some sense? And they had to be frozen, okay, because that makes it a lot easier to deal with it rather than put in formalin or other kind of ways to fix tissue, okay, so it had to be frozen. It had to be pretty good size, and that was because we were going to run it across each of these platforms, and that just requires sheer amount. It couldn't be necrotic. That means that dying cells, it couldn't be more than 20% necrotic or dying cells. So we had all of these stipulations. So what does that take us to? That means that one of the limitations, although powers, of the TCGA is that every single cancer, whether it was lung, breast, you name it, with the exception of cutaneous melanoma, had to be a primary tumor. It was just the rule. And the reason for that is that to get large enough samples, really, in most cancers, you can only get them from primary tumor in a reproducible way because of lung dissections, breast dissections. And in uveal, if you think about it, that's true as well, from enucleations. So all of the samples were primary enucleated samples. The only exception would be if we had an enucleated sample and a metastasis from the same patient, and we really tried to do that, and we didn't get any that passed QC, okay? So it's an important point. 80 enucleated samples, and that was because of all the stringencies of frozen, large enough, of the QC, okay? So when you step back from this, I can just even ask the audience, what do you see? What does this group compare to this group? What's the obvious difference? Again, blue means you've gone less than 2 for chromosome whatever number, and red means you've gone more than 2. Is this a less chromosomally affected set of tumors than these? Yes, right? These are almost all normal. The only recurrent was really this 6p. Go here. This 6p gain was pretty common. Now, some of these things, as was said earlier, right, Dr. Rios, was that some of these things had been bubbling up through the literature by lots of groups working really hard. The benefit we had is that we had the state-of-the-art sequencing technology to do this. So we could really be confident in a lot of our observations. But as you cut across, so group 1, this is somatic copy number alteration group 1, that's the only recurrent aberration, 6p gain. Group 2, you get the 6p gain, you get a little bit of 6q loss, you get a little 8q gain, but it's not even the full Q in a lot of cases, varying amounts. The more red, the more increased number you have. Now, 3 and 4, the obvious thing is a loss of 3P, a loss of 3Q. We call that monosomy 3. And so what was interesting, our first observation was, well, the monosomy 3s are getting cut into 2 different groups. Even though they're all monosomy 3, they're getting cut into 2 groups, and it looks like it's based upon these aberrations. This 6Q loss in about half of these guys, the 4s, an 8P loss in many of these, and a very high 8Q gain versus a lesser 8Q gain. And so if you just look over here, I put this in today just to emphasize the point. Again, monosomy 3, you lose 3P, 3Q. You just have 1 chromosome 3. And in the case of a gain, you get a whole bunch of gains of 8Q, and some losses of 8p, all within the abnormal tumor cell, okay? And each of these, of course, has genes on them. And so these genes are in greater number. Doesn't mean they're greater expressed. but they're greater number. And obviously the genes over here are less, and we know about the BAP1 story. BAP1 was the only recurrently altered gene on the remaining chromosome 3. We looked at every other gene, no other recurrently mutated one, just BAP1. Very different than a lot of cancers. So that's the chromosome. So in the chromosomal aberration analysis, we end up with 4 groups, splitting disomy 3 into 2 groups, splitting monosomy 3 into 2 groups. Now, integration. Now we look at, please don't get overwhelmed with this, now we look at the methylation. As I mentioned earlier, you can have on-state and off-state. In this case, yellow means that that particular gene and its methylation status, if you cut across, that's a specific gene, and each column is a sample of the 80 samples. So 80 samples going down, and each gene cutting across. So this very first gene looks like it's a lot more hypermethylated, and then it loses that methylation, and then it's hypermethylated. And you can keep going down each gene. Well, again, you step back, you let the computer line things up. What are more like each other than other things by methylation, only methylation, and it perfectly aligned with chromosome 3 loss. So if you lose chromosome 3, you have this methylation pattern. These genes are very methylated. These genes are much less methylated in comparison to the other disomy 3, okay? This was a first. Nobody had actually shown this, that the loss of BAP1, monosomy 3, followed by BAP1 mutation, which is how it happens in time. We actually figured that out too. So you lose one chromosome 3, you get a BAP1 mutation on the remaining chromosome 3, and that's highly associated with this very distinct methylation pattern, indicating that this process of methylation of a whole bunch of different genes seems to be very important for whatever BAP1 loss does. And that leads us down a specific trail of both research and also therapy, because we have things, drugs, that can modify the methylation of genes. There are FDA-approved drugs for hypo and hypermethylators. And so I'm not saying that we have the one in hand, but it now takes us down that path because of that observation. So the summary thus far, monosomy 3 have a 1-to-1 overlap with BAP1 alterations. Monosomy 3 tumors 1-to-1 overlap with a specific DNA methylation profile. Monosomy 3 and disomy 3 tumors are each separated actually into 2 types, 2 subtypes. Now back to genes. So a lot of work had been done, a lot of wonderful work by Boris Bastian and Bill Harbour and many other groups identifying mutations in GNAQ and GNA11. And for a good amount of time, everybody heard the same mantra. 90% of uveal melanomas have a gene mutation in either Q or 11. They're mutually exclusive, one or the other. And that left that 10% a little open. And then over the last few years, and including our study, we find that all uveal melanomas, I mean, there's always going to be an exception where maybe we missed it, but 99% of uveal melanomas have a mutation in either this gene, cis-LTR2, about 4%, PLC beta 4, about 1%, or GNAQ, GNA11. And the math here is actually a little off. This should be a little higher. This should be about 3%. This should be about 5%. And this, but the idea is 99% will have, and they're mutually exclusive. If you have a mutation in this, you do not usually have a mutation in these, vice versa for these. And that makes biological sense because this gene is a receptor and codes for a receptor. It's one of the phone numbers that gets sent, and a protein in the membrane of the cell that connects normally to GNAQ and 11. So it made perfect sense that if you're mutated in that one, you wouldn't have a mutation in the remainder because they use the same pathway. And we've seen this in other cancers. In cutaneous melanoma, if you have a BRAF mutation, you don't have an NRAS. If you have an NRAS, you don't have a BRAF. Why? Because they're in the same pathway. It's redundant. So the same redundancy happens in uveal melanoma with these 4 genes. And that's why you rarely see overlap. And when you do see overlap, it tends to be an oddball mutation. in, let's say, Q versus, But anyway, so those are the important ones. And again, a lot of people have done a lot of work to look downstream of these, and including my group looking at, can we block MEK? Can we block RAF? Can we block PKC? These pathways that are clearly downstream. And I think it was described earlier, and well described earlier, that it's a bit of a whack-a-mole problem still. I hit MEK, but guess what? Junk, GNK, comes up. Literally, yeah. And so you ever play whack-a-mole? You hit the mole, another mole pops up. Now you can hit that mole. Now, don't be discouraged, because in some other cancers, they've figured out how to triangulate and hit the right moles in unison or in combination or in parallel to shut down pathways or to enhance the immunotherapies. So we're working our way at that, but again, having a comprehensive understanding of what the mutations are. And we think these are all very early mutations. These mutations probably exist in nevi, just like BRAF mutations in cutaneous melanoma exist in nevi in the skin. They're sufficient to create a small nevus, but they're not sufficient, we don't think, to go on to metastasize. So they're a first hit, and they're universal across uveal, it seems, one or the other, okay? And just to show that graphically in the paper, we took those same 80 tumors, and you look at 11Q, I call it CYS, PLC, and you look across the CYS, there's no Q or 11 or PLC beta. Same here. This one's interesting, but you'll notice it's green. And why is that? Because that GNA 11 and GNA Q was a 183, not the 209 you hear about. 183, weaker activator. And thus, it is capable of having also a PLC beta. But this PLC beta doesn't. And so that's that mutual exclusivity, except in rare cases. And the rare cases tend to be odd cases. Okay? So 1 of 4 genes is mutated. We think it's early on initial event. Then it's followed by other mutations. Either a mutation in SF3B1, a mutation in IFAX, or a mutation in BAP1, and these tend to be mutually exclusive. Not always, a little less so than the others, but they tend to be. They co-occur with the other 4, by the way, so don't get me wrong. When I say mutually exclusive, I mean with each other, these 3. They co-occur with the other 4 I mentioned. And so we already learned about BAP1 and when that gets mutated after a monosomy, and these guys tend to get mutated in the disomy state, when you have 2 chromosome 3s, okay? So they tend to be associated with a better outcome because they're not associated with monosomy 3, BAP1. But I gotta be careful because, you'll see in our 4 groups, right? 2 disomy groups, 2 monosomy groups, you see some overlap a little bit with the SF3B1s and BAP alterations. The bottom 2 rows are BAP alterations that we discovered by 2 different methods. And there's some overlap, and some of you in the crowd might be saying that to yourself. Hey, I had a BAP mutation and I had an SF3B1, and that would be this case here, okay? But the vast majority of IFAX mutations, EIF1AX, are in black over here, happen in this setting when you have a very low changes in your chromosome. Remember that far column where there weren't many chromosomal changes at all? Those are the ones that tend to have IFAX mutations. That's why you might hear people say, well, if you have disomy 3 and you have an IFAX mutation, and you are probably in a very good outcome category. Then the SF3B1s, you can see almost always, with one exception, and again, it's an oddball, it's a blue, not the more common red, are in this group, which correlate with this chromosomal state, a little more chromosomal aberration going on. And so these can metastasize, but they tend to have a bit of a different character. It's still being worked out, but there have been publications that have shown they tend to occur a little bit later. They tend to be a little slower in their course. They tend not to necessarily have liver tropism or involvement, although they certainly can. But our data was shown through the TCGA was showing that there's actually, it looks like there might be 2 different ones. And I already showed you how they're different in their chromosomal states, but they look like they're also different in that the subset 3, we'll call it, of monosomy 3s, they are more apt to have the SF3B1 mutation. And then we discovered a new gene to be mutated, and it's just in the same family. It's a very similar gene. In fact, you know, it has a lot of S's in it. You know, so SRSF2, and we discovered 3 samples, sorry, that had mutations in those, and they tended to go with the SF3B1s. And so, and subsequently now a few other groups have found mutations in this family of genes which are, have a splicing function I won't go into, okay? So this kind of, again, comprehensive, integrated analysis is really revealing a lot of interesting observations, connecting mutations with certain copy number and then certain methylation patterns. I should have mentioned that too. The IFAX1 mutations had one methylation pattern, if I went back to that yellow and blue diagram. So again, that methylation pattern seems to be a very good methylation pattern if you're going to have uveal, that one. So to further summarize in my Furhter Summary, GNA11Q, these 4 genes show a mutually exclusive mutation pattern. They happen early. These 3 genes show a mutually exclusive pattern for the most part. They happen later. They're second hits. We discovered a new gene, and there's a few more that have now been discovered. They're very much like the SF3B1. IFAX mutations align with very specific copy number and methylation patterns. SF3B1, same thing, kind of specific copy number and methylation patterns. And I didn't show the data, but that had been shown by another group prior to us, is that we don't see the classic UV mutations that we see in cutaneous melanoma, in eye melanoma. And on average, we only see 30 mutations total, and those are the drivers, the ones I showed you. Those are the ones that are activating or having a functional effect, I should say. Those 7, now 8 that I showed you. And so that's, if anybody's kind of not made this connection, one of the thoughts with why cutaneous melanoma is so sensitive to immunotherapies is that in the early days when they opened up a cutaneous melanoma tumor, they said, what are all these lymphocytes doing in here? What are these immune cells doing in here? They were recognizing the tumor. And now we finally have the drugs to push them along to not only recognize it, but finally attack it. When you look in uveal melanoma, both in the primary and in the metastatic setting, Dr. Patel did a great job of showing this in both primary and metastatic tumors. They're not as highly infiltrated. By about 10-fold less, they're not as infiltrated. So automatically, there's a difference, it seems, either in the recognition or in the activation or both. And so understanding why that's the case is a big part of what many groups are doing now. And it won't just help uveal melanoma and many other cancers that have a very low mutation rate. Why do mutations matter for the immune system? Because these mutations happened after you were born. And so they're foreign. And there are skin melanomas that have 6,000 mutations, and they're all foreign. They weren't there when you were born. They're there in the tumor. And so that's why skin melanomas are so immunogenic compared to uveal, at least in part. It's much more complicated, but that you can think of it that way, foreignness. And so we're working on figuring that out. I won't even go into this. I know, this is where it stops. You're like, it's got enough. This is it. Enough of this colors. But all I wanted to point out is that I did mention to you that we were going to look at these features, mRNA, lincRNA, mirRNA. And all you have to do is take the 50,000-foot view and say, look, they also grouped into groups of 4. And so there was just incredible overlap between the mutations, the copy number status, the RNA statuses within these tumors. And that's something that's it's a clue. It's telling us that these are programs, very clear programs, and understanding where to put the right default in these programs is our task for sure. But if we do go up maybe 10,000-foot view on mRNA, okay, again, 80 samples across, each column is a sample, across is a gene, the mRNA of a gene, and these are the genes, 12 genes that make up the class 1, class 2 distinction. that you all are aware of. And so it turned out that we were separating the high-risk category, it looked like. Our cluster 3 and cluster 4 were actually separating the high-risk category. And interestingly, the one that was associated with the cluster 4 had a greater immune infiltration. And even though it was low level, there is a separation in primary tumors between ones that have immune cells, which is this red just represents high level of, versus blue, low level of in here, these genes. And so some of the tumors did have a level of an infiltrate in them, that the immune system was apparently recognizing them. And then there were some that were just stone cold. Okay? And learning more about why that's the case is what we're trying to do. So if we look at Disomy 3 and Monosomy 3 in our cohort of TCGA, it looked exactly like every cohort that's come before us, in that if you were Monosomy 3, this is time, and this is a drop from 100%. And this is a Kaplan-Meier curve you saw earlier. And this is the one we don't like to see, where over time you see patients getting metastasis. This is to get metastases and getting metastases over time, versus the Disomy 3s, much less in their frequency. And what was interesting is that methylation pattern that I showed you was 1-to-1. We could have used that as a marker instead of chromosome 3. It was that clear. But what was also interesting is that Monosomy 3 group split into an early versus later to onset of metastasis group. And so that we're very interested in, because one criticism, clear criticism, is, well, maybe you just, caught them later and earlier. You might have just, and maybe that's all that the biology you're seeing, this big difference between Monosomy 3s that are subset 3 and Monosomy 3s that are subset 4. Maybe that's all the difference you're seeing, is the biology of a later tumor versus. But it didn't match up with size or any of the other factors really well that we looked at. It's only half of 8, it's about 43 tumors total. So we can't say a ton more, but what we can say at least is that, boy, they really did seem to separate in their time to metastasis, and I won't go into it, but they really had different biologies, not only in all the features I showed you, but when we do pathway analyses and really look at how these things map onto things we know about how cancer works, quite different. Now, you don't want to get ahead of yourself, but one thought is, well, maybe if it truly isn't representing the fact you're just getting, to a later onset tumor versus an earlier onset tumor, perhaps the biology in this primary tumor may represent some of the biology in the cryptic, not yet seen metastatic tumor. Maybe the environment that allows for these two biologies is similar to, in some important way, the environment in the liver. So let's cross-compare. And so that's what we're doing now. We're saying, how much are the liver mets like this? How much are we learning from this about the liver mets? And that sort of integration between primary and metastasis is a major focus right now. And with that, I'm going to just end with this as the model. So the model we come up with is that you have a group that has IFAX mutations. They have a very different DNA methylation pattern. They have a very parsimonious, not highly aberrant chromosomal status, only the 6p gain. I didn't go into their molecular programs, but they have some specific molecular programs, and those tend to have a very good prognosis. These are probably your class 1s, these are the things that groups measure with for prediction. The SF3B1s tend to have also a DNA methylation pattern that overlaps with them. Not 100%, but not too bad. A little more copy number alterations, but not the Monosomy 3. And the 8Qs are not so, are not as, the 8Qs are not as many in copy number. And then they have a different program I didn't go into. And these tend to look like they have an intermediate prognosis. And then the Monosomy 3s with the BAP1 losses, they have a very definitive DNA methylation pattern. The caveat was mentioned earlier, all enucleation. Does this correlate with smaller tumors, non-enucleated tumors? And that's being asked at this moment by a few groups as well. And so that's an important question. Does it correspond with metastasis? Important. And then the most important thing I wish we could get a handle on is for that tumor that you can't see yet, is it more like this? Or has it deviated a lot? And so we're going to actually, at the end, learn the two ends of the spectrum, because that's what we're going to have tissue on. And I can tell you that, at least by mutation and copy number, the Mets don't look all that different from the primaries. So if there's some other biology going on, it's epigenetic. It may be other things that we did, features we didn't test, but we did a pretty good comprehensive, integrated analysis. And with that, these are the take-home message. Advocacy really matters. I mean, this would not happen except for the bubbling up. The next project wouldn't happen if we didn't push this hard. The UK group and the French group are part of the European group that was mentioned earlier. They've used this because they were part of it to say to their folks, hey, we're doing great work. And this all starts to intersect. Comprehensive integrated investigation, you're tired of those two words. Investigations require highly functional, skilled teams. This was a huge team approach, and a lot of people willing to work together, hang their egos at the door, and that's what really matters. And I would say that we pretty much have identified all the gene mutations and chromosomal aberrations in primary. I don't know how anybody could get deeper than we've gotten. There's always space for more, but that space is narrowing, narrowing, narrowing. is a cancer whose features, like I mentioned, mutations, chromosomal aberrations, are highly overlapping. I think we're learning a lot about these programs. And high risk may subdivide into two groups with different characteristics. And with that, this was the paper that was published. Over 30 authors and over 170 collaborators, and that doesn't even count all of the great work that was done at each individual institution from people who didn't make it on the paper. So we're talking about a colossal, wonderful group effort. And then I do want to point out a few people. The first author, Gordon Robertson, a computational biologist and data person from the Canadian, British Columbian Cancer Group, phenomenal work. Juna Oba works in my lab. She was like the glue. She just kind of kept all these parts together and kept it moving. Surya Kendoth from Memorial Sloan Kettering was my co-chair. A lot of the computational work as well. Bita Esmali from MD Anderson, who just was smart enough many years ago to just start putting tumors in a banked frozen. And then many years later, we have the right application, you know? And so thinking ahead. And with that, I hope, mine says 15 minutes I have. If I'm sorry if it was a bit of a tour de force, but hopefully it gives you some sense of the kind of research that we did.
Question, Dr. Woodman. It seemed like earlier I heard you say, if you don't have the BAP1 mutation, there's a different course toward metastasis.
I would qualify. I mean, I would say that we need to do more SF3B1 mutant, or non-BAP, I should say as a broader category, non-monosomy 3 BAP1 mutant, okay, tumors tend to have a different course, although it's not been studied as well as we need to study it. There are folks who have a very similar course, but on average, it tends to be a bit of a different course, at least among the SF3B1 mutant patients without a BAP aberration that have been evaluated. Is that because there's less of liver involvement? Maybe it's more subcutaneous or involves other tissues, and therefore, you know, these are the things that need to be better understood. It's not that they don't get metastases, of course they do. They tended to be later after primary, and again, we have to really follow these patients out in a large number, but they may have a bit of a different course on average,
Okay. Is there any data that's been produced on the time span from primary diagnosis to metastasis?
That's, so those Kaplan-Meier curves where you see that's exactly what these curves represent here. So this is 100% or 100%, 80%, 60%, 40%, right? And this is time, and this is from diagnosis here of primary uveal melanoma to the time that uveal melanoma specific metastases, that is metastases not from something else, was diagnosed. And so you get to the 50% mark at, in this case, in the 40-somethings months, which is about, these curves are pretty similar to what's been published many times over for monosomy 3, disomy 3.
Okay. Where does it go after 5 years?
No, you're seeing the curve here. You end up, every tick mark these little tick marks here represent a patient who was lost to follow-up or who is censored, meaning that they don't count at that particular point. And then every drop in the curve is due to a percentage, of patients from 100% going down to 20%. And that it ends there because the last one, as you could see, was censored. So doesn't drop to 0. Yeah. I'm sorry. So blue is monosomy 3, and red is disomy 3. So disomy 3 was 37 patients, ultimately having 2 events. And monosomy 3 was blue, 33 patients, ultimately having 14 events. And I forget, each time you show a Kaplan-Meier, you should say how long you followed folks for. I don't remember in this case, we didn't have the follow-up that a lot of other folks do because it was a cohort that was pulled together from multiple different places. But this is, if you look at the literature, this is how we represent following events. Some event, it can either be the onset of metastasis or it can be survival in time from an original group based upon some characteristics. In this case, it was the characteristic or the feature was DNA methylation. In this case, it was the copy number. In this case, link RNA, mRNA. So by those different features, you could see different rates. These were the same rate because, as I mentioned, monosomy 3 and DNA methylation pattern were 1-to-1. So they're the exact same patients. In this case, it splits this group, the monosomy 3 group, splits into 2 groups. As you can see here, 19 patients, 6 of whom had an event, 14 patients, 8 of whom had an event, and this represents a difference in their time to metastasis. And so if the biology, again, a small number, granted, but deeply characterized samples showing a difference in their time to metastasis. But you have to be careful in these cases and say, well, there's 19 patients and 14 patients followed for only X amount of time. So, yeah. Okay, did that answer your question, sir? Okay.
So these all came from patients with enucleation.
Correct.
How much of that is required? Where are we at with respect to the ability to classify folks according to all the data you've got here now? And if you've had the Castle genome done, can it go back and be reclassified into this?
It's a great question, and what we'd have to do, I haven't approached Castle. I have talked to Bill Harbour, who came up with this 12-gene test. It would be very interesting if they had residual RNA, from these, not these specific patients, because we don't have that, but from like patients, if you would, and whether or not you also see this difference in they're both. If you had enough RNA, you could look at different genes that are like you can tell these genes are very different from these genes in their expression profile, right? These are very low, these are very high for specific genes. So you might be able to come up with a further sub-classifier of the poor prognostic group. They actually did that for the good prognosis group. It was a 1A, 1B subdivision, which actually maps very well to the subdivision that you saw in our Disomy 3 group. The one thing about the castle that is been published multiple times as being a beneficial feature is that some of their class 2s don't have Monosomy 3 or BAP1 alterations, and they think that they're capturing a certain feature potentially of the environment of the tumor that is unique, and that might be a bad prognostic tumor environmental aspect. But definitely, these are the things that we'd love to keep looking at. I think the insights from this are that's what it was meant for. You know, we put the paper out there, all of the data is publicly available, and other groups can now, you know, see how their data correlates with ours and start looking for the things that we've observed.
Thank you. This is incredibly exciting stuff, just in case anybody thinks it's boring. What I really came here to talk to Sarah about, and this is a good time now, is really about treatment, who it's working for, finding out the characteristics of those people and mapping it back onto this, so we have a much better idea as to which treatment to give to which metastatic ocular melanoma patient.
I couldn't have said it better in the sense that one of the programs that at Anderson, not just in uveal, actually it's been more in the skin melanoma patients, is we call them exceptional, you know, responders or non-responders, you know, and so folks who are just breaking all the rules by being a standard deviation, 2 standard deviations right down, and so we just published actually in JCO Precision Oncology a patient like this in skin melanoma with a KIT mutation where, you know, this patient has been alive on 2 small molecule inhibitors for 5 years, and we just don't see that, right? And so we did exactly this, the deep drill down, the comprehensive integrated, you know, and we identified that she looked to have features that allowed her, based upon the treatment she was on and what we knew about what targets those drugs were hitting and the features of her tumor, that luckily she just got on the right phase 1 trial that was hitting the very other aberration that was the resistance feature. And I think it's exactly right. We learn a lot by going deep on those 1, 2, 3 very exceptional cases. And the hard part is quite honestly to get the tissues, you know, and to process them properly and to store them properly and then to do the right. I mean, all of that, I was trying to stress that, you know, because it is so imperative, but it's very hard to do. It's hard to fund. It's hard to have an infrastructure that allows for it when you have a busy clinical operation going, you know, and it requires a lot of patience, you know, on the patients to take that time, to take the, you know, that slight extra risk of a biopsy. You know, all of those things have to come together. But the more educated we all are on the risks and benefits, I think, you know, again, these things start to interconnect and we gain strength through these parts.
So one quick comment and one question related to what you just said. It seems like we have 2 of those unicorns in the room with us today that could be studies as to why they're still here. I would like to see that happen. I know that Nancy volunteered for that. But my
Can I just make one point real quick on that? Is I know that there's a registry. There's always been a hope to get a registry. I know that's more well-formed. And that would be the kind of thing that would be an important column in a registry is, do you have prior biopsy tissue of any kind? Where is it? You know, how could we, you know, MTA, material transfer agreement, to get a hold of it and build a study around it? You know, exactly that. Okay, I'm sorry.
That's okay. My question is, I know that uveal melanoma, OM, is a melanoma, but it's so different from cutaneous, is there any other type of cancer that this has similarities to?
It's a great question. So BAP1 loss has been identified as being a somatic aberration, and it means not from born with it, right? In renal cell carcinoma, about 20%. I might get my percentages off here, but renal cell, mesothelioma, cholangiocarcinoma, and then in smaller percentages of many other tumors. And so BAP1 loss biology is a space in cancer, just as much as you might have heard of P10 or P53 or RB1 or all of these other tumor suppressors, quote unquote. BAP1 is just as legitimate a tumor suppressor as any of those, and it's now been found in multiple cancers, and then it's also now even germline, as you all know. You're at risk of getting all the ones I just mentioned if in a family if you have a born with the mutation in that. So if we better understand BAP1 biology, I think that's going to be a key insight, and we can potentially do that through other cancers.